All right, today we're going on an incredible journey. Not across the world, but deep, deep inside the human body. We're going to peel back the layers and explore the absolutely stunning microscopic world of our tissues, the hidden fabrics that literally make us who we are.
So, let me ask you a question. What are you really made of? I mean, we think of ourselves as a whole person, right? Arms, legs, organs. But what happens when you zoom in? And I mean way in. The answer is so much cooler than just cells. It's all about how those cells get organized.
Well, it looks a little something like this. Just incredible. It's this beautiful, highly organized community of trillions of cells, all working together to form what we call tissues. And you can think of each tissue as its own unique fabric, with a very specific job to do. The study of these fabrics, that's called histology. And today we're going to see exactly how scientists make this invisible world visible.
So here's our game plan. First, we'll look at how we can even see these tiny structures. Then, we'll get into the 4 fundamental fabrics of the body. And after that, we'll take a closer look at 2 of them, the protective linings and the supportive framework that holds the whole show together.
Okay, so what exactly is histology? Well, to put it simply, it's microscopic anatomy. It's the study of our body's tissues. It's the science that lets us see the fundamental structures that are the basis for how our organs work. And honestly, it's the foundation for understanding pretty much everything about health and disease.
But here's the catch. You can't just take a piece of fresh tissue and stick it under a microscope. It just doesn't work. It's too thick, it's way too fragile, and honestly, it's mostly transparent. So, to see anything, we have to transform it from this living, squishy tissue into a permanent, visible slide. And that is a really fascinating 4-step journey.
First up is fixation. You can think of this as basically pickling the tissue. We use chemicals, usually one called formalin, to preserve it and stop it from decaying. Next is embedding. Here, the tissue gets infused with paraffin wax to make it nice and firm. It's kind of like encasing a strawberry in a block of Jell-O before you try to slice it. Then comes sectioning. This is where we slice that hardened block into these incredibly thin ribbons. And finally, the most important part, staining. This is the magic step where we use specific dyes to add color, making all those invisible details finally pop.
And there it is. This is what you get after embedding. That little piece of tissue is now safely locked inside a solid block of wax. This gives it the support and the rigidity it needs for the next step, which is just unbelievably precise.
Okay, so meet the microtome. This thing is essentially a super high-precision deli slicer for tissue. It shaves off sections that are only about 5 to 10 micrometers thick. Now, how thin is that? Well, a single human hair is around 70 micrometers thick. So yeah, these slices have to be insanely thin so that light can pass right through them.
So, we've got our perfectly preserved, ultra-thin slice of tissue sitting on a glass slide. But we still have a huge problem. Most of the components of a cell are totally transparent. So how on earth do we actually see anything?
The answer? We stain them. And this is where the science gets really, really cool. We use different dyes that are chemically attracted to specific parts of the cell. For example, a basic dye like hematoxylin loves acidic things, so it stains the cell nucleus a deep purple-blue color. Then, an acidic dye like eosin stains the basic parts, like the cytoplasm, a vibrant pink. This contrast is absolutely crucial. Pathologists literally use these color patterns to diagnose diseases by looking for tiny abnormalities in how the cells take up these stains.
All right, so now that we know how we can see these tissues, let's talk about what we're actually looking at. It might seem impossibly complex, but believe it or not, every single structure in your body, from your brain to your big toe, is built from just 4 fundamental types of tissue. That's it, just 4.
This table gives us a great overview of those 4. Now, I want you to look at that middle column, amount of ECM. ECM stands for extracellular matrix. That's basically all the non-cellular stuff, a mix of fibers and fluid, that surrounds and supports the cells. And this is a huge differentiator. Look at epithelial tissue, very low ECM. The cells are packed tight. Now look at connective tissue, high ECM. It's mostly matrix with cells scattered about. This one difference tells you so much about what they do.
And wow, you can really see that difference right here. On the left, you've got epithelial cells packed shoulder to shoulder, almost like bricks in a wall. They're built to be a barrier. But then on the right, look at that connective tissue. The cells are sparse, kind of just floating in that vast sea of extracellular matrix. This structure isn't for lining things, it's for providing a strong, flexible scaffold.
And here are the other two to complete the set. On the left, you have muscle tissue. See those long, aligned fibers? That structure is perfectly engineered for one thing, pulling in one direction to create movement. Then, on the right, you have this intricate branching network of nervous tissue, which is designed for communication, for sending signals across the body in an instant. These four fabrics, they are the building blocks for absolutely everything.
Okay, so let's do our first deep dive. We're going to start with epithelium. I want you to think of this as the body's interface tissue. It lines every single surface and cavity. It's the boundary between us and the outside world, and it also separates all the different compartments inside our bodies.
So, what makes epithelial tissue, well, epithelial? It's all about its structure. The cells are packed together super tightly, with almost no extracellular matrix between them, which lets them form a continuous sheet. They line everything from your skin to your entire digestive tract. They also form glands that secrete all sorts of important stuff. And one more thing, they always rest on a thin layer of connective tissue called the basement membrane, which acts like an anchor.
Now, speaking of glands, they're actually formed by epithelial cells that dive down into the tissue below. And they come in two main flavors. You have exocrine glands, which stay connected to the surface through a little tube or duct. Think sweat glands or salivary glands. Then you have endocrine glands. They lose that connection to the surface and instead release their products, which we call hormones, directly into the bloodstream.
And if we look even closer at those exocrine glands, you see they can produce different kinds of things. On the left, we have serous cells. They make a thin, watery secretion that's packed with proteins, like the digestive enzymes you have in your saliva. Then on the right, you've got mucus cells, which, as the name suggests, produce a thick, slippery mucus that lubricates and protects surfaces. And there's a third major type you should know about, lipid-secreting cells. You find these in the sebaceous glands in your skin, and they're responsible for producing the natural oils that keep your skin and hair waterproof and moisturized.
What's really fascinating is not just what these cells secrete, but how they do it. This shows three different ways. With merocrine secretion, little vesicles inside the cell just fuse with the outer membrane to release their contents. Your pancreas does this. With apocrine secretion, a whole piece of the cell membrane actually pinches off. That's what happens in mammary glands. And then there's holocrine secretion, where the entire cell basically fills up with product and then self-destructs to release it. That's how our oil glands work. Pretty wild, right?
Okay, let's shift gears. We've talked about the linings, now let's talk about the stuffing. Let's get into connective tissue. If epithelium is the body's wallpaper, then connective tissue is the structural framework of the house. It's the studs, the foundation, it's everything that supports and connects all the other tissues together. And like we said before, the biggest defining feature of connective tissue is that it's the complete opposite of epithelium. It's all about that abundant extracellular matrix, that mix of fibers and fluid. It's the matrix that gives each type of connective tissue, from bone to cartilage to even blood, its unique properties. And that's what allows it to provide both structural and metabolic support for the entire body.
So, think about the journey we just took. We went from a piece of raw, living tissue all the way to a beautifully stained slide. We've explored the body's fundamental linings and its supportive framework, and what we've seen is that at every single level, structure dictates function. Which leads us right to the core purpose of histology, learning to read the stories that these fabrics are telling us. Because by understanding their normal architecture, we can learn to recognize the subtle, tiny changes that signal disease, turning this incredible microscopic world into one of our most powerful tools for diagnosis.